SeeSAR is a very intuitive and visually enabled tool for the medicinal chemist to bring out ideas into the active site and test different ligands directly in situ with respect to their estimated affinity and other important ADME-tox properties. When a PDB file is loaded, we see the protein structure in 3D together with the co-crystallized ligand, and a click in the table reveals a more detailed picture about the binding situation. This detailed picture is visually communicated by putting so-called affinity coronas on each atom, which tell the chemist directly, without looking at any numbers, how well each atom contributes to the affinity. A green corona means the corresponding contribution from this atom is good, and a red corona means there is a penalty. The size of the corona corresponds directly to the amount that this atom is contributing to the affinity. The chemist can now decide to edit a molecule. In order to do that, we need to select a binding site first. And the ligand needs to be brought into the ligand tab and can now be edited. When the editor is open, we can engage various helper tools, such as the quality of the conformation of the ligand. We can turn on the so-called unoccupied space in the binding site, which shows us the tightness of fit for each ligand. So when we see fog, this fog around the ligand, there's space for at least a fluorine atom to be added. So for example, we can now add a methyl group here. The editor is open above the molecular table, and we can change this H into a carbon and see directly the influence that this has on the affinity. So in order to compare this to the reference, we need to calculate the reference as well. This is the co-crystallized ligand. And we can see that this edit basically maintained the current estimated affinity for this particular ligand.